So hello, I'm Jeff Banka, and I'm going to be talking about Module 2, Improved Plumbing, looking at the evolution of plants and paleobotany in deep time. So 430 million years ago or so, we see the first vascular land plants. These are plants that developed these vascular tissues. These are water and nutrient conducting tissues. This is versus 470 million years ago that we see the first land plants. So there's about a 40 million year gap or a lag, something 40 to 38 million years ago, or 38 million years. And here's the geologic time scale here, looking at deep time, where we're talking about millions of years in the past. And what this illustration by Ray Troll and Kirk Johnson in one of their books depicts is what we call the Phanerozoic eon, which basically comprises the Paleozoic, the Mesozoic, and the Cenozoic, the three major eras of multicellular life on Earth. So harking back to how do you create basically a land plant from an algae, an aquatic algae moving onto land, you basically are forming several key innovations. And some of these, when you first invade land, or developing a cuticle that's hydrophobic, keeps you from losing water, developing basically stoma or stomata, or ways of breathing through the cuticle. So having more active or more direct gas transit and water vapor transit, then coating your spores in basically a hydrophobic shell or encasing or spore wall that will help contain a micro offspring or propagule of a plant, so it can be wind transported across the landscape. Now, the major innovation that we're going to talk about today in this module is basically, if you wanted to create a larger plant or become a larger one, I speak many times when I say I, because I like to put myself into the mind of a plant, which doesn't have a brain or a nervous system, but bear with me. I like to think they behave. In terms of if you wanted to become a large plant and you envision yourself on this rocky, barren landscape that has no shade invented yet, and blazing hot sun, lots of UV, lots of wind, you're going to want to stay close to the water. You don't want to lose your water. You don't want to get dehydrated. You don't want to trek out into the desert, which is most of the geologic sort of lichen landscape, if you will, at that time, because those are probably your only friends, or lichens at this point. To get bigger, you're going to have to develop water and nutrient conducting tissues. They'll be specialized, and we'll talk about that a lot more soon. So about 430 million years ago, we see the first plants to develop the advance of these specialized water and nutrient conducting plumbing through their systems. Our little fellow here on the right cartoon sort of exemplifies that. You can see those little tubes running. That's basically as if it was made of glass. You can see the water and nutrient conducting tissues going through it. And it basically functions like a straw. It sucks water out of the ground to bring it up and transport it to tissues that are further away from the ground and more complex and large, versus just using basically osmosis, where you're pressing one thin layer of cells against the ground and just absorbing water through capillary action, more or less. So this is what will need to happen. So we see the first instances of this in Cooksonia, basically the earliest known land plants that are vascular. And Cooksonia are basically like little tuning forks that basically green tubes, tuning forks that have spore-bearing capsules we'll talk a little bit more about shortly, but they lacked roots and they lacked leaves. So they were photosynthesizing through these axes, these little stems they were putting up. But what does it mean to be the oldest vascular plant? Well, vascular plants are united by a number of different traits. The three major criteria, if you're working with the fossil record and going from a sort of geological forensic investigator's perspective, is you're going to look for these three things. You want to see the presence of vascular system, and that is basically any cells that are specialized for transporting water and nutrients and assimilating products basically throughout the plant's body, moving chemicals and such defenses. The next thing you also want to look for in the fossil record is presence of a cuticle with stomata, because stomata, these specialized pores that often have two guard cells that basically will open, swell, and close to moderate water loss, are often associated with the piping of this vascular system, having a lot more water. Being able to suction water out of the ground requires there to be not only plumbing that goes down into the ground, but also basically where the suction's coming from, the atmosphere in a way. So the plant has to regulate how water is moving up and out of its system. And the way to do that is by opening and closing these little stomata that allow gas, basically transpiration and water vapor to come out, and fuels this suction further down the plant. It's a fascinating topic, very complex. It's very much, Mission Impossible, but plant style. Trilete spores were another trait that you see in the fossil record that's associated with many of these vascular plants. And it allows for reproduction in an environment that is quite hostile. We're talking about this windswept, UV blasted, irradiated landscape that it's not friendly for a little aquatic algae. So you want to be able to have a spore that has the right kind of shell, or I keep saying shell, it's a spore wall that is impermeable and is also has UV deflecting pigments on it, so it can transport itself through that nasty environment that we call land and the air. But its role is to transport genetic material as the next generation further inland, in this case. So here we have another one, a chart here looking at our time. So geologic time, going from the bottom to the top, we're going from older to younger. The Ordovician, the middle Ordovician period is our earliest land plants. So our first step is in the evolution and the invasion of land, as we call it with plants, is you have a bryophyte domination stage. And this lasts for quite a while. Then you have a second stage, and that is the ecological change and the increase in vascular plants. And these are the plants with that sort of plumbing we've been talking about. Above, we see different evidence for different sorts of fossils in the geologic record, ranging from traits to actual presence of certain types of plants or groups of plants that we're about to talk about. So the first vascular plants, I'm sorry, we should say that here, is about 430 million years ago. And it's this Cooksonia plant we were talking about to the very left on the screen. You can see that schematic of what we call the dichotomizing branches. We're then going to talk about some more of its buddies that come around a little later in the fossil record. So one of the early sorts of traits that we see are the trilete spores. And these are what we were just saying, these three-ridged or tri-ridged spores that are specialized for moving through the air and keeping the spore dry. That's our first evidence of a vascular plant right there. And that's as far back, it's looking like about 460 million years almost. But we see the first definitive records about 430 million years ago with Cooksonia. So between the appearance of these sorts of trilete spores and the appearance of definitive fossils of Cooksonia, we have about 38 million years of evolution going on. It took a long time to get this sort of trait. 40 million years ago was a long time ago from today. So if you imagine that time span of going back, the dinosaurs died out about 65 million years ago. So if you imagine much of the age of mammals it took for plants to get past this trait up to the diversity of a Cooksonia, it really gives you an idea of how difficult of an environment this must have been to live in, and how hard it was to actually get these traits actually to develop. So looking at the cast of characters here, or our usual suspects in fossil record, we are basically starting with Cooksonia, going through this series here. This is the occurrence in the fossil record chronologically of when the first appearance of each of these major plant types or plant groups shows up. And we're not going to address these in the same order here. We're going to talk about basically how they work in terms of complexity. So more about that soon. So our first stage, or our first, basically our first phase of vascular plant evolution, starts with a group called protracheophytes. And they don't really have a very good common name, so we just call them protracheophytes, which means before tracheophytes. And tracheophyte is the technical term for vascular plants today. So one of the examples that we have here is called a gleophyton. And a gleophyton is much like what we were talking about with Cooksonia. It is this dichotomizing or tuning fork branching sort of plant. It has no true roots, but it does have rhizoids, these unicellular sorts of water conducting cells that grow into the ground. You see in many mosses, liverworts, and I think hornworts have them too, but don't quote me on that. The spores are produced at the very tips of the branches in structures that are called sporangia, these capsules, and they get their sporangia high up off the ground so they can cast them off the environment. A gleophyton is really interesting because it also harbors fungi, symbiotic fungi in its tissues that infect through, I think the rhizoids or such, where they're making contact with the ground. But if you think of a looping little runner on the ground with little tuning fork like branches, you get a good idea for Cooksonia. The main thing about them that we want to bear in mind is that they have no tracheids, specialized water conducting cells, but they do have a less specialized conducting element that you can find associated with their fossils. And here we can see more of Cooksonia here. These on the left, these are scanning electron microscopy images of spores down at the bottom. And they're trilead, as we can see. They have 3 ridge arms. They may look a little contorted, but I mean, I don't think I'd look that good after 430 million years myself. But above that, you also have a scanning electron microscopy image of a sporangium. This is the structure that carries and produces the spores. And you can see that it basically is like a capsule, almost like a bit like a salt shaker in a way, when it's cracked open. So they basically had these very simple vascular systems. They were branching in twos dichotomously, no roots, but they have rhizoids, and they don't have any leaves. So they're photosynthesizing throughout these branching axes. Here we can see a few more of the traits they have. Cuticles. They do have cuticles with stomata in this case. So they've already developed those. They do have the trilate spores, and they do have somewhat of a vascular system, but it's just not quite as specialized. There are some things that we call Cooksonia that are a little more advanced than other Cooksonia. So I should say that Cooksonia is a name. It's sometimes regarded as a bit of a waste bin for some enigmatic fossils that have these tuning fork, fragmentary branching, stick-like fossils. Some of them were larger and more complex than others, and others were earlier in the fossil record, look like they haven't developed all of these traits. So within what we call Cooksonia as a group of plants, there's a number of these traits showing up in some of the members. So it's a lot of evolutionary sort of experimentation going on in this group. So the next group I want to talk about in terms of complexity is zostrophils. And zostrophils are certainly more complex than Cooksonia in terms of now they have subterranean underground shoots in many cases. They have a vascular system that is more complex typically. So they have very specialized tracheids and cells for conducting water nutrients. They also have developed small spines, some of them, to help with photosynthetic area. They still don't have true roots and they still don't have true leaves. But they also have these sporangia that are no longer limited to being just the tips of branches. They're actually arranged in the axils, basically. They might be associated sometimes with spines, but they grow lateral sporangia, which now they could pack more spore-bearing capsules on a single branch. So that was quite an advance for reproductive gain. And you can see in these images here, these clusters, they're not quite cones yet, but they're a little bit like you're aggregating different little spore-bearing capsules like a pod. The next, really, in terms of complexity, we get the trimerophytes. Many of these probably gave rise to the ancestors of our seed plants and the ferns and horsetails. They do have usually a central stem, so they have what's called a pseudomonopodial way of growing. So they dichotomously branch, but they have a dominant shoot with each division instead of a tuning fork. They grow much larger. They get their spores much higher off the ground. They still don't have any leaves, and their sporangia are also terminal, but they branch a lot more than the Cooksonia. These here at least also would produce roots. So roots evolved in the trimerophytes as well, but still no leaves. And now I want to bring your attention to a group called lycophytes that today are represented by club mosses, coolworts, and spike mosses. There was a group, this group had one representative very early on called Baragwanathia, and it was a bizarre and remarkable plant in that it was gigantic, and it had huge leaves, true leaves that were long and narrow. It's been interpreted that those were basically developed, some of them developed sporangia in association with them as well. And Baragwanathia it's interpreted to be one of the early forerunners to our modern lycophytes. But we're talking about, Cooksonia might be a few centimeters tall. This plant's the size of a timber bamboo. Why that's important is because of the order of appearance in the fossil record. So if we look at all these plants I just showed you, we were talking about levels of complexity as we moved further and further. Turns out that basically the rhinophytes, Cooksonia, and its relatives come earliest, which makes sense. But suddenly you have Baragwanathia showing up, like well over 400 million years ago. Out of nowhere, you have suddenly this plant with full-size leaves, the size of a timber bamboo, living in a world full of micro 2-centimeter tall plants that haven't even evolved vascular tissues yet, to some extent. So when we look at the increasing complexity, it doesn't make any sense why Baragwanathia would be found when it was found. And it was found initially in Australia. The original specimens were found in Australia. Paleontological community kind of balked at the results because they're like, well, this must be a fluke. This must be the wrong dating. Turns out that Baragwanathia shows up as you get high latitudes in Canada at around the same time. It's also showing up in the northern hemisphere too. So we can say that, it's not a site-specific thing. It actually is a very early plant. So it's one of the great mysteries how that occurred sort of out of order in a way. But it shows that the fossil record is notoriously incomplete, and there's many clues that we may or may not ever find out there that may have been preserved. So looking at the improved plumbing, we see that now, as we look toward the right of this family tree, we're getting into some of the more modern vascular plants, what we call the tracheophytes. And the modern vascular plants have a deep division, the lycophytes and the euphylophytes. So Baragwanathia and the rest of its lycophyte kin diverged along with those zosterophils, with those lateral sporangia. Those form the sister group, or the other branch to all the other modern vascular plants. And the lycophytes evolved their roots and their leaves independently of all these other plants that are vascular too. So the trimerophytes evolved their roots separately, but Baragwanathia had its leaves for dozens of millions of years longer than any trimerophyte before the first leaves emerged in trimerophytes. So lycophytes basically beat euphylophytes to the punch on almost every evolutionary innovation other than flowers and seeds, which they never evolved. So it's a very interesting thing when you look at a modern selaginella in a garden and you think, well, that's been doing what it's been doing in ways for 400 million years. So with that, I'd like to conclude module 2 with a few take-home points. So spore assemblages basically show an increase in vascular plants across the landscape early in the evolution of plants on land. So about 400 430 million years or so ago. The vascular plants basically can be divided into two major groups, or what we call clades: the lycophytes and everything else, which we call the euphylophytes, which includes today our horsetails, our ferns, our horsetails, ferns, and our seed plants, which includes the gymnosperms and the angiosperms, the flowering plants. And the other thing really to take into account is with this deep divergence, it's almost baffling to think that lycophytes developed leaves and roots far earlier and independently of all these other land plants. And we see the same evolutionary events happening in the other ones much later. So I like to tell people that when you think of lycophytes versus euphylophytes, a lycophyte might be like an early land plant you threw onto Mars and gave 400 million years to evolve. And it did everything similarly, but it did it a little differently. So it's one of the fascinating things that always draws me back to these plants. So without further ado, I'd like to open it up for questions.